Okay, so we're going to start talking about patterns of inheritance. Most of our conversation is going to be about Mendelian inheritance, recessive and dominant traits, and how they are passed down through single genes. But some of the conversation will also be about complex polygenic traits. For example, many genes govern metabolism and blood sugar, so it takes a lot of them going wrong to give you, let's say, type 2 diabetes. But let's start with Mendelian or single gene inheritance. It deals with monogenic traits. A gene codes for a protein. If there's variation in the alleles, which are the types of genes that code for that protein, you get some variation. Mendelian inheritance does not address complex polygenic traits. You can't use a Punnett square, for example, to predict diabetes in offspring very easily, but it can predict the risk of occurrence and recurrence of a monogenic disorder. And we do that with knowledge of a pedigree and Punnett squares, and I'll show you the Punnett squares today. Alleles are variation of genes. Alleles vary by very small sequences of DNA that have different nucleotide sequences. So not to get you too lost, but every 3 bases makes a codon. A codon codes for an amino acid. You get enough amino acid productions, you will get a protein. The sequence of how they are bonded makes a difference. So any change to those codons in the DNA can make variations in the type of protein that's formed. Most of the time that's benign. Sometimes it impacts your health. When you inherit the same allele from both parents, it's the exact same sequence, you are considered homozygous for that gene. And your genotype, your DNA, will match your phenotype, which is your appearance. If you inherit 2 different alleles, there's variation among the bases, you are heterozygous for that gene. And alleles typically express their traits as either dominant, which for the most part always expresses itself when it's present. For example, type A blood, person who has the gene that creates the antigen for type A that attacks people with type O blood, have a dominant trait. It always expresses itself. We represent dominant traits with capital letters. Recessive traits remain silent if a dominant allele is present and are only expressed if all genes are recessive, and they are represented by lowercase letters in this example B. Okay? What if different dominant alleles combine? For example, you have an allele for type A blood, and it codes for that A antigen, and your spouse or your mate has type B blood, and they code for an antigen. Your offspring could have type A blood. Both genes will express themselves at the same time. You will have the A antigens and the B antigens. Okay, that is codominance. Incomplete dominance is a little bit different. We have 2 dominant genes, and let's say in this example that your mom had straight hair and she was homozygous. She had 1 dominant allele. Your dad had curly hair and he was homozygous, dominant. All their offspring will have wavy hair because the traits sort of meet in the middle. They combine and have a blended phenotype. Okay, so here's how Punnett square works. You may have seen this in other biology courses, or you may not have. If it's new information, there's more about it in your book. But all it is, it's a very simple grid. And for each trait that we're looking for, we know that each parent has 2 genes. So in this example, we have the maternal axis is vertical and the paternal axis is horizontal. We put 1 letter in each spot, and then we kind of go across and down. So in the first block, in the upper left, mom donates a dominant allele, dad donates a dominant allele. In the second block, the upper left, mom donates a dominant allele, but dad donates a recessive allele. And that offspring is heterozygous. Then we can go to the next block, which is in the lower left. Mom will donate a recessive allele, dad donates a dominant allele. That child is heterozygous. And in the last block, the lower right, both parents donate their recessive alleles and that child displays the recessive trait. Now, it's important to remember, and you've probably already heard this, that we're calculating probability. We're not saying that if these people have 4 children, that 2 of them will be heterozygous, 1 will be, because, I mean, you think about it, a woman is born with 1 to 2 million ovum. Men create about 1 million sperm every day, or is it 300 million, that can live for 72 hours. So this is, think about it in terms of research. This is a really tiny, tiny, tiny sample of a very large population. And so it's the probability each time. It's like flipping a new coin. Okay, so if mom has, let's say that this is for eye color, and the dominant allele is brown-eyed, and the recessive allele is blue-eyed. She could have 4 children, and all 4 of them have brown eyes, all 4 of them have blue eyes, 3 have blue eyes. Any combination is possible, but each time you have the same probability. Every pregnancy has the same probability of producing this outcome. All right, and that's how Punnett squares work. As we move along with monogenic inherited disorders like sickle cell disease and cystic fibrosis, we'll be doing the Punnett square to show you how that works. Okay. Genotype, phenotype, it's a very simple idea. We all have 2 sets of alleles. Some alleles will express their traits and others don't. The genotype includes both alleles, whether they're dominant or recessive. The phenotype describes the traits that are expressed. When your genotype is homozygous for a certain trait, your phenotype will match the genotype for that trait. When it's heterozygous, the genotype is different from the phenotype because you're hiding that recessive gene. All right, so we're going to talk a little bit about autosomal inheritance, and then we're going to take a little break so this video doesn't get too long. There are 22 autosomal pairs, right? The first 22 chromosomes are called autosomes, and they can transmit traits in an autosomal dominant or recessive pattern, or exhibit codominance or incomplete dominance, or one of those. But alleles are likely to be transmitted to males and females equally because the sex chromosomes work a little bit differently. For example, here's our brown-eyed trait again, capital B, and here's our blue-eyed trait, lowercase b. And we do the same Punnett square. Mom gives a brown-eyed gene, dad gives a brown-eyed gene. Mom gives a brown-eyed gene, dad gives a blue-eyed gene. The phenotype is brown eyes, but the genotype is heterozygous, has both dominant and recessive. And we did the same thing down here. Okay, so that's kind of how that works. That's autosomal inheritance. It's pretty simple. Some conditions that have an autosomal recessive pattern of inheritance, we have some benign traits like blue eyes, lack of tasting, PTC, that's a chemical that's found in like olives and chocolate and coffee, and some people perceive it to be bitter. Earlobes being attached to your head is a recessive trait. Red hair is a recessive trait. Left-handedness are recessive traits. So you can go several generations and not see a red head, and then boom, there's one because of recessive genes. Same thing with left-handedness or blue eyes. You might have 3 generations of brown-eyed parents. Blue eyes came from somewhere, but they were sort of hidden behind dominant genes the whole time. Then we have diseases. And most of them are a lot less common than the traits we're talking about here. So we have sickle cell disease, in which it's inherited as a recessive disorder. Now, it's very common in certain racial and ethnic populations, African Americans, of course, but also some Latina people and Middle Eastern people also tend to carry that trait more commonly. Tay-Sachs disease, which is common in Ashkenazi Jewish families, as are many other disorders, cystic fibrosis. Some of these genes confer an advantage if the person is heterozygous for the trait. For example, sickle cell trait is thought to protect to some extent against malaria. Cystic fibrosis was thought to protect against cholera. But these are some disorders: beta thalassemia, hereditary hemochromatosis, albinism, all those. Now we have dominant patterns of inheritance. Benign traits: brown eyes, freckles, tasting PTC, having brown hair, having right-handedness, having an A-positive blood type. Those are all benign traits, and they are inherited in a dominant way. And you could work out a Punnett square for any one of them and figure out the probability of an offspring if you know what the genotype of the parents is. Then we have diseases. Now, I'll kind of discuss why some diseases survive despite the fact that they are inherited in a dominant pattern and occur in every generation. Achondroplasia is a type of dwarfism. It involves shortened limbs, but normal head size, normal intelligence. Folks usually function fairly well. Most of the time, it comes out of nowhere in what's called a de novo mutation. You don't have to know that yet. Someday it will be important. A de novo mutation is one where the base is changed for the very first time in the parent's sex cells. Once they are in the cells of the offspring, they can be passed down. Marfan syndrome is another example of an autosomal dominant pattern of inheritance. Many times it is caused by de novo mutation, but it can be passed on. Huntington disease, autosomal dominant, but does not occur until most people have already fathered or given birth to offspring. Neurofibromatosis, also dominant, can be very mild, where you have some cafe au lait spots and some benign fatty tumors that do not cause trouble. It can be very devastating, where the tumors press on the spinal cord and the nerves. Long QT syndrome and sudden cardiac death syndromes tend to be dominant, but they don't usually impact the person's health until they are older and have had a chance to reproduce. Malignant hyperthermia, it's an interesting one, and any of you that are going into operating room, labor and delivery, any place where you might have to see general anesthesia, you're going to see the CRNA or the anesthesiologist ask about problems with anesthesia. It is dominant, but you would never know you had it unless someone you know was exposed to certain anesthetic agents like succinylcholine. But it is inherited in an autosomal dominant pattern, and it's important to get that history before the person receives any type of anesthesia. And then we have hereditary non-polyposis colon cancer, HNCC. It's one of those familial cancers, and again, the person usually does not, the trait does not start expressing itself right away. It switches on later in life. Same thing with Huntington. And so sometimes a disorder that's passed on through autosomal dominance survives, the gene survives because people reproduce before they know they have it, or they reproduce having a mild case, and because of a phenomenon called variable expression, meaning the severity varies from person to person and is not dictated by the presence of the gene, they might reproduce and not realize that their child could be severely impacted by that disease. Okay. We're going to stop here, and we're going to pick up again with the patterns of sex-linked inheritance, because I think that's really, really important to understand. A lot of really devastating childhood diseases are inherited through the X chromosome. So we'll stop here, take a little break, and then we'll pick up again.